Hello, and welcome to Wellness Wednesday. I'm Erin Kuhn-Krieger from the Rolfe Pancreatic Cancer Foundation, and I'm here tonight with a raw and heavy heart. Yesterday, the Rolfe community lost one of our own to this horrible disease, and I lost a friend. He was a devoted husband, dad, son, brother, and friend to so many. He served on Rolfe's board and was a fierce advocate in sharing his story to raise awareness and raise funds for early detection research. He's left such a profound impact on me and each of us who knew him. He will be greatly missed, and we're holding his family in our hearts tonight. I know I'm here tonight knowing that I'm not alone in this feeling. I know so many of you have felt the same way about your loved ones, and that's why we're here. We're here for the hope. There's so many researchers out there dedicated to progress and finding better outcomes for pancreatic cancer patients, and Rolfe Foundation seeks out and provides funding to those showcasing the best possibilities for breakthroughs and change. It's because of these breakthroughs and those to come that so many lives are going to be saved. Tonight, we'll hear from Drs. He, Bissonnette, and Macleod from the University of Chicago. U of C is just one of the leading institutions that receives research funding from Rolfe. They're also our partners when we need to find immediate care, often within 24 hours, for Rolfe families in crisis. For more information on what we fund and how you can help, please visit rolfefoundation.org. Before I introduce the doctors, I want to mention that you could ask questions throughout tonight's session by putting them within the chat box. I'll be holding all questions until the end of the program to make sure that we're able to get to everybody. And if you don't feel comfortable putting your questions in the chat, you can always email them to us at info@rolfefoundation.org. Tonight, we have three speakers. Dr. Marc Bissonnette is an associate professor in the Department of Medicine, section of gastroenterology at the University of Chicago. Dr. Chuan, the current John T. Wilson Distinguished Professor at the University of Chicago and an investigator at the Howard Hughes Medical Institute. And Dr. Kay Macleod, a research professor and program leader of molecular mechanisms of cancer at the University of Chicago Comprehensive Cancer Center. I'll let each of them share more about their background and experiences. So first, I'd like to introduce Dr. Macleod. Welcome, doctor.
Thanks, Erin. Before I start, I just want to add my condolences on the passing of Erin Colwell. I think it's tragic passings like this hopefully will spur us on and renew our determination to find better ways to diagnose and treat pancreatic cancer. So first of all, I want to thank the Rolfe Foundation for supporting our research this past year. The work I'm going to talk about this evening relates to a very specific aspect of pancreatic cancer called cancer cachexia. So as I mentioned, it is a feature of pancreatic cancer. In fact, pancreatic cancer is the type of cancer most linked to cancer cachexia, to muscle wasting. 85% of all pancreatic cancer patients present with cachexia. And unlike other cancers where this is generally a feature of late stage disease, with pancreatic cancer, over 50% of patients who present with early stage or earlier stage and locally advanced cancer already exhibit cachexia. So it starts earlier in pancreatic cancer than it does for other cancers. What is also true is that the amount of weight loss that patients experience predicts disease outcome in pancreatic cancer. The more weight lost, the shorter the prognosis, the worse the prognosis. And you might be surprised to realize that, in fact, about 30% of pancreatic cancer patients actually die from the cachexia that causes heart failure. The heart obviously is a muscle as well, and muscle weakness, and not the tumor itself. And currently, there is no medical intervention for cachexia. So this is where we have focused our research interests in the last year and a half to hopefully, make a contribution to better understand what is causing cachexia and to hopefully mitigate against it, to block it. This diagram I show here kind of summarizes what we think is going on, where the tumor growing in the pancreas is releasing signals of some kind, and what those signals are, we do not know, that circulate to the muscle and cause the muscle to break down. And then the breakdown products from the muscle are thought, perhaps, although again, we really don't understand, to somehow promote the growth of the tumor. So in looking at cachexia, there are some fairly well-established markers of cachexia, of muscle wasting, including these two genes here, atrogen-1 and MRF1. These are both proteins that are known as E3 ubiquitin ligases. These are enzymes that are involved in degrading protein and contribute to the degradation of muscle. We looked at this, and this is both in human patients and in mouse models, and together with work from other people, we have shown that this gene BNIP3 also appears to be upregulated specifically in the muscle of cachectic animals and patients. And as you can see here, the full change in BNIP3 is actually over 100-fold greater than what you see for these two other established cachexia genes, or atrogenes as they're called. So question, what is BNIP3? BNIP3 is a protein that is found primarily at the mitochondria, the powerhouse, the energy factory of the cell, and it plays a role in promoting the turnover of mitochondria through a process known as autophagy. This involves the formation of these double membrane structures that engulf mitochondria to form a double membrane vesicle that fuses with the lysosome, which contains degradative enzymes that degrade the mitochondria, resulting in the release of amino acids, nucleotides, and lipids that allows the cell to continue growing and surviving. So BNIP3, which is the focus of what I'm going to talk about tonight, and its role in cancer cachexia, functions at the mitochondria in this manner to promote the turnover of mitochondria by autophagy. So this, building on the model I presented earlier, led us to propose that the tumor growing in the pancreas is releasing a signal that circulates to the muscle to upregulate BNIP3. We do not know what that signal is, but one of the things we're focused on is understanding what is inducing the upregulation of BNIP3 specifically in muscle. This then leads, at least in our hypothesis, to increased mitophagy in the muscle, causing degradation, further degradation of the muscle, and release of metabolites and possibly other molecules that circulate back to the tumor to promote tumor growth. So the work I'm going to talk about, present here, is the work of two talented young ladies in the lab, Georgina Mantinelli, a postdoctoral scholar, and Elizabeth Foster, who's actually an undergraduate at the University of Chicago. And this work has been, as I mentioned, kindly supported by the Rolfe Foundation. So we generated a model of cancer cachexia. First of all, we genetically engineered this mouse model in which BNIP3, this gene of interest in cachexia, is specifically knocked out or deleted in the muscle as a result of breeding it to this muscle-specific Cree strain. I don't want to go into the details, but this is a completely novel and unique tool that we developed for our studies. What we were intrigued to see, and consistent with our hypothesis that indeed BNIP3 was involved in promoting cachexia, is if we just look at muscle atrophy in this mouse, in this case in response to overnight fasting. You can see here on the left, the control fed mice have big, juicy muscle fibers. But after fasting for 24 hours, you can see that the fibers have got a smaller cross-sectional area, and there's a lot more space between the muscle fibers. In contrast, when BNIP3 is knocked out, there is no atrophy. The muscle appears to remain intact, and there's no loss of mass. So this was preliminary data that we were on the right track, but obviously this is just fasting, and that is not what we were most interested in. We went on to cross these muscle-specific knockout of BNIP3 to a pancreatic cancer model. In this model, the KRAS oncogene, which is the key driving oncogene in human pancreatic cancer, is activated, and P53 tumor suppressor, which is one of the main tumor suppressors in pancreatic cancer, is inactivated. When we looked at the effect of knocking out BNIP3 in the muscle of these mice with pancreatic cancer, we were, first of all, validated, as we anticipated, that BNIP3 is induced in these mice with pancreatic cancer. You can see this increase in BNIP3 expression, but when BNIP3 is not knocked out, as you would expect, we do not detect its expression. More interestingly, we see decreased expression of this atrogene, this gene that we know is driving cachexia, or one of the genes driving cachexia, that is decreased in expression when BNIP3 is knocked out. In addition to this, we took the mice that had developed pancreatic cancer and started to monitor muscle strength, clearly important in terms of the muscle decay, how muscle decays, both in these mouse models, but also in patients. Amongst one of the assays we perform, and this is not the only assay, but one that I'm going to talk about this evening, we measure grip strength of the mice using this device here, which allows us to assess the muscle tone and the muscle strength of the animal. Other assays involve putting the mice on treadmills and measuring respiration rates of the mice. But bottom line, the key finding here is that deleting BNIP3 in muscle protects the muscle from both atrophy, but also maintains the strength of the muscle. Again, in support of our idea that BNIP3 is a key target for preventing cachexia. When we measured muscle area, this is the cross-sectional area of muscle fibers. Again, here you can see, again, these studies are still ongoing. It's a relatively small number of mice here, but we are accumulating more data every week that's consistent with this, showing that knocking out BNIP3 in the muscle of these mice with pancreatic cancer protects the cross-sectional area. Again, you see the mice are maintaining the cross-sectional area of muscle fibers. And this is unrelated to overall body mass. This is very specific to the muscle. So this was, confirmed our hypothesis, and we were happy and relatively excited to make this finding. What was even more exciting was not just that loss of BNIP3 in muscle was maintaining muscle mass and strength, but it actually also, as you can see from this graph, was also causing the tumor to grow less. So this is a key finding, and one that's completely novel in the field, the idea that you can affect tumor growth or pancreatic tumor growth through effects on muscle and BNIP3 expression in the muscle. So this was a really very exciting finding for us this past year. So this led us to our hypothesis I mentioned before, where are now more validated by the data from these mouse models, that BNIP3 is driving cachexia in the muscle. Again, there's this two-way traffic from the tumor to the muscle, and then from the muscle to the pancreas and the pancreatic tumor. And that by inhibiting this process in muscle, we're actually stopping the supply of nutrients or other molecules to the tumor, and thereby inhibiting the growth of the tumor, in addition to maintaining the tone of the muscle in these animals. So at the moment, one of the things that we've started looking for is assessing the plasma from these mice and looking for differences in circulating metabolites that could explain how the muscle is supplying the tumor with pro-growth nutrients or other molecules. Also, we're looking for alterations in circulating cytokines that potentially could be inducing BNIP3 in muscle. So again, we're looking at this two-directional traffic, both in terms of what is the muscle doing for the tumor and what is the tumor doing to co-opt the muscle into serving the tumor, the growth of the tumor. So obviously these are mice. We use these mice as essentially a discovery tool. But obviously in the long run, our goal is to leverage what we find in these models to understand what is happening with cachexia in pancreatic cancer patients. And for that, our work, we're lucky in being at the University of Chicago, which has a very a long-standing and a very deep tumor bank of not just pancreatic tumors, but also, interestingly, I was delighted when we found this, that we actually have this resource. The doctors here have had the foresight over decades to collect muscle from patients at the same time as they're resecting pancreatic tumors. And also we have serum from patients. So we will be able to hopefully validate whatever we find in the mice as driving the induction of BNIP3 in muscle, and following on from that, whatever the muscle is producing to feed the tumor, we'll be able to validate that by looking at these patient samples, both the tumor, the circulating blood, and also muscle. The key question then, of course, because obviously our genetically engineered mouse models use genetic tools to inhibit BNIP3. In patients, we need to find a biochemical approach to inhibiting BNIP3. The question is, how can we do that in patients? And this is where I return to this MRF1 gene, which, as I mentioned, is an E3 ubiquitin ligase that is upregulated in muscle of pancreatic cancer patients. And so we want to exploit this enzyme to degrade BNIP3 using this approach, which has become it's a recent approach that's been developed in a number of different labs. But this is a unique application of this technology, which is called PROTACs, where we engineer an artificial peptide linking, in our case, the transmembrane domain of BNIP3, which will bind to BNIP3, link it via peptide to a ligand that is normally recognized by this E3 ubiquitin ligase. And what that will do will result in the proteolytic degradation of BNIP3 at the proteasome. We're essentially hijacking this muscle-specific enzyme, which is upregulated in cachectic muscle, to degrade the protein that we think is driving the whole process. And so this approach, again, we're working with our colleagues in chemistry to develop this approach, but this is our longer term goal in terms of leveraging our findings from the mouse model to apply it to human patient treatment and outcomes. So with that, I'll stop and I'll take questions at the end, as Erin mentioned. But it's now my pleasure to introduce my colleagues at the University of Chicago, Dr. Chuan Hu and Dr. Marc Bissonnette in the Department of Medicine. Chuan and Marc.
Thank you all. I don't know if you can hear me or not. I'm a gastroenterologist at the University of Chicago and very interested in pancreatic cancer and colon cancer, and I've been grateful for the Rolfe Foundation and for an opportunity to discuss this with you. So let me see if I can get my screen shared here. So briefly, I'm going to tell you about our research that I've been doing in collaboration with Dr. Chuan He to make a diagnostic test to diagnose pancreatic cancer at earlier stages and potentially even pre-malignant pancreatic lesions, and to distinguish those that are more likely to go on to cancer versus those that are less likely to do that. Oops. That didn't work. Sorry. to advance the slide here. Okay. So as many of you know, pancreatic cancers are an increasingly serious problem in the United States. They're thought to be perhaps the most serious malignancy as a cause of death in the next 30 years. And so it's a rising incidence and it's a serious problem. There are several pre-malignant precursors of pancreatic cancer, including the pancreatic intraepithelial neoplasms or PANNs and the interpancreatic mucinous producing lesions, the MCNs and the IPMNs. These lesions are thought to progress over time, and we're interested in developing a blood test that would differentiate pancreatic cancers from normal or at least non-malignant lesions, and eventually to be able to detect pre-malignant lesions as well. DNA circulates from the tumor and from all organs of the body into the bloodstream and carries with it many signals that can be used for important diagnostic tests. One of the important bases in DNA is cytosine. Cytosine can be methylated in the 5 position to make 5-methylcytosine, or can be hydroxymethylated in the 5 position to make 5-hydroxymethylcytosine. In the first part of the talk, I'm going to talk about this particular residue, 5-hydroxymethylcytosine, that Dr. Chuan discovered and created an elegant way of assaying in the blood. These modifications are quite stable and they carry important information because DNA coming from the tumor has these modifications that can be detected in the bloodstream. Basically, the DNA from diseased tissue, from pancreatic tissue, is released into the blood, and using a technology that Dr. He invented, the nano-HMC seal technology, he has been able to capture these residues and detect them in the blood. Previously, this required a very large amount of DNA that was beyond the amount that circulated in the blood. So this was a really novel and exciting test that he developed. So blood is taken from the patient that carries important DNA signature from the blood of the cancer. The 5-hydroxymethylcytosine is first conjugated to a glucosyl residue, which contains an azide molecule on it. This can be captured by another molecule called biotin with click chemistry, and then the biotin can be brought down with streptavidin beads, which allow it then to be amplified and sequenced to look to see throughout the genome where these 5-hydroxymethylcytosines are occurring. Because as I mentioned, in cancer, they're altered from the normal state and can be detected as such. So if this is an example of a gene, a normal gene that has 5-hydroxymethylcytosine labels on it, in the cancer, there's an increase in the number of the 5-hydroxymethylcytosines in some genes, as an example in this gene 1, whereas in this gene, there's actually a loss of 5-hydroxymethyl, so there's a lower number. And so the point of the test that Dr. He is working on with us is to develop a way to measure these differences up and down of 5-hydroxymethylcytosine that could be used as a diagnostic signature for the cancers. They also discovered that there's a relationship between the 5-hydroxymethylcytosine and the gene expression. So each tissue has its own unique sort of expression levels of genes, which essentially cause that tissue to be what it is. In the case of pancreas, a unique organ for pancreas. And there's a relationship between the gene expression levels shown on the y-axis and the 5-hydroxymethylcytosine levels shown on the x-axis. And when that was examined in relationship to other known markers of gene transcription, for example, the acetylated histone 3 lysine 27 protein, which is associated with genes that are undergoing active transcription, there was a close association with increasing amounts of 5-hydroxymethylcytosine and increasing amounts of this marker in the histones. And this has been a finding that's been verified by numerous other labs, that 5-hydroxymethylcytosine essentially marks genes that are undergoing active transcription. So basically the test begins with blood from the doctor's office. The DNA is isolated on silica columns. PCR adapters are placed on the DNA in order to be able to PCR amplify it up later on. The 5-hydroxymethylcytosines are first labeled with a glucosyl molecule, and then those are reacted with biotin that binds to the azide on the glucose moiety. The biotin is then captured by streptavidin with beads that are coated with streptavidin, and then the DNA with the PCR adapters is amplified and sequenced to give rise to where then looking throughout the genome, one can look to see how the 5-HMC signature has changed from normal to a cancer patient. We had a great opportunity recently. We applied and were granted access to samples from the PLCO study. This is a study that was initiated back in 1993, PLCO for the diagnosis of prostate, lung, colon, and ovary. And it was to test whether all the screening tests that were done at the time, chest x-rays for lung cancer, flexible sigmoidoscopies for colon cancer, CA-125 and vaginal ultrasounds for ovarian cancer, and PSA and digital rectal exams for prostate cancer would be protective against these cancers. And I won't summarize the numerous findings from the study, but suffice to say that these very valuable samples taken from 155,000 people who were at the time free of cancer, and then they did serial blood samples for 5 years and then have followed them over time. And out of that 155,000, 400 individuals were to go on to develop pancreatic cancer. So we have blood samples from patients prior to their clinical presentation, which may serve to validate the ability of the test we're working on to diagnose early pancreatic cancer. So this is we've been also doing our own prospective studies, collecting samples from patients at the University of Chicago and also from California with Dr. Goyal's help. And in our own patients, we have about 80 patients with stage 1 or 2 colon pancreatic cancer, about 15 patients with early pancreatic lesions that are prior to the pancreatic cancer. They're comprised mostly of patients 60 to 80 years old. They're predominantly male, although we have 40% of them female, and the race is predominantly Caucasian, but we have a substantial number of African Americans as well, about 15% of the population. And the analysis that we're working on now will incorporate not only the 5-HMC distributions to make the diagnosis in the blood, but it will also look at factors such as diabetes, obesity, tobacco, and alcohol that are important risk factors for pancreatic cancer. So this is some of our preliminary data. In the first panel, we're showing pancreatic cancer patients here in blue. Each column, if you will, represents a separate patient, and then controls are shown in green. And each row represents a different gene in the genome. And we've measured the 5-HMC expression levels of those genes by next generation sequencing, and this is in the blood now. So what you see is if the expression of 5-HMC is low, the gene is pseudo-colored blue. If it's high, it's pseudo-colored red. And so in this set of genes, in the controls, there's relatively lower 5-HMC, as denoted by the blue color, compared to the patients with cancer. Their 5-HMC in the blood is higher. And conversely, there are other genes in the blood and the controls that were higher in 5-HMC, whereas the cancer patients had lower 5-HMCs for those genes. And if you do a further analysis by principal component analysis, it looks at the statistical distance between these 5-HMC loci in the genome, you can show that most of the cancer patients are segregated to the right here on this analysis, as opposed to the controls to the left. So this is a test that I think ultimately will be very useful to be able to make a blood diagnosis of pancreatic cancer, but we still have work to do, obviously. Then we turn to the pre-malignant lesions, things like the Ipmn lesions, and we compared those to control patients versus the Ipmn patients and did a similar analysis in the blood now for 5-HMC circulating. And again, the high levels are denoted by red, the lower levels by blue. And again, we had another signature that distinguished the control patients with low 5-HMC for these genes and higher 5-HMC for these genes, and similarly another group of genes that had higher 5-HMC for the controls and lower for the Ipmns for the pre-malignant pancreatic lesions. And again, if you did a principal component analysis, again, you found that the controls were to the right, and in this case, the pre-malignant lesions were to the left. So that although there's some overlap, we think that as we get better with this test and incorporate some genomic features in the tissue, that we'll be able to have a test that will diagnose pre-malignant lesions in pancreatic cancer. And this is a similar analysis comparing pancreatic cancers in blue to pre-malignant lesions in orange. And again, we have a limited number of patients, but there's a sense that there will be a separation, a tendency of separating the pre-malignant lesions in blue from the pancreatic cancer shown in red. We then turn to the tissue. All the studies I've showed up to this point were blood studies, which is the most important tissue in terms of making diagnoses and following risk factors. But we also wanted to look at the tissue because there are probably important signals of 5-HMC in the tissue itself of the pancreas, which may help us guide selection of those we focus on in the blood. And so for these studies, we compared normal pancreas to the Ipmn lesions in the pancreas for 5-HMC. And again, we found that there was again a signature that in patients with normal pancreas, there was relatively low 5-HMC expression for these genes compared to high expression in the Ipmn lesions, of the pre-malignant lesions in the pancreas. And conversely, other genes had a decrease an increase in the normal control and a decrease in the patients who had pre-malignant lesions of the pancreas. And what's even more important ultimately would be to be able to distinguish those that are high-risk lesions that are pre-malignant in the pancreas from those that are low-risk lesions, so-called high-grade versus low-grade dysplasia. So in a preliminary small number of patients, we looked at that for 5-HMC signals. These are again now in tissue now, not in blood. And we're looking at high-grade lesions, which have about a 70% chance of progressing to pancreatic cancer within 10 years, and comparing it to low-grade lesions that have about a 20% chance of progressing to pancreatic cancer. And again, you see in the 5-HMC signals in the tissue, there are some genes that are upregulated in the more dysplastic tissue with respect to 5-HMC expression compared to the less dysplastic lesions on the right. And conversely, there are other genes that have a downregulation of 5-HMC in the tissue that are at higher risk to progress. And these tissues have in the control case, they have a higher expression of 5-HMC. So we think that ultimately comparing the tissue and the blood will be useful, and I'll come to that in a moment. Right now, with the help of the City of Hope Comprehensive Cancer Center and the University of Chicago, we've continued to accrue more samples. We're working on developing enough patients for a training cohort so that we'll have a good signature of the things that I've showed you before, of these 5-HMC levels being high or low relative to controls. And then we'll once we have a signature based upon the pancreatic cancer and the control patients, and our controls incidentally will include chronic pancreatitis, which can be a confounder in this whole field because these patients present with pain similar to pancreatic cancer. They have abnormal CAT scans and abnormal imaging. And so we're hoping to use them as a good control to develop a test that will distinguish those patients from the pancreatic cancer patients. And we also have, of course, the other valuable samples that I mentioned earlier, the PLCO biorepository that will have at least 200 samples from patients who went on to present with pancreatic cancer, to compare those to age and gender matched patients who were followed over many years and never developed pancreatic cancer. So ultimately, we want to integrate the 5-HMC signals from the blood and from the tissue, because we think that the combination will enhance the identification and prioritization of disease-specific features that are captured in the cell-free DNA. So the test will ultimately be a blood test, but we think that the tissue will give us some guidance to make it an even better blood test. And as I mentioned before, these pre-malignant lesions come in two varieties: those that are thought to be at high risk to progress. Those would be, for example, the main duct Ipmns that have about a 70% chance of progressing, and those that are thought to have a low risk of progression, such as the branch duct Ipmns. And once these have been identified if we have a blood test that would allow us to characterize whether they are high risk or low risk, we would know how often to surveil these patients and how careful and how soon we might need to move towards surgery. So for the risk stratification training, we're also doing studies with City of Hope, where we're collecting plasma from high-risk patients with these pre-malignant precursors, as well as blood from, sorry, in the wrong direction here, as well as blood from patients with low-risk lesions and healthy controls. And we're also getting tissue from these patients that we'll be able to use for genomic typing of the 5-HMC in the pancreatic tissue to compare to the blood 5-HMC signatures in the blood. So our options ultimately for this test will be to continue with the next generation sequencing based assay, which I think is quite strong and a very good assay, or to develop some sort of a PCR based assay for a limited number of loci that are identified as part of the signature. And in that case, it would be more translatable to the clinic. And in that case, we would do the same sort of workflow where we'd take blood from the patient, we would isolate the DNA, we would selectively label the 5-HMC, we would use click chemistry to pull it down, but instead of using next generation sequencing, we would do PCR to amplify up the loci that we've identified as either being upregulated for 5-HMC in these genes in the circulation compared to controls, or to be downregulated in the 5-HMC compared to control. And if we could identify a number of loci that would show this sort of a differential expression of 5-HMC up or down, we think we might be able to make a very rapid assay that would be affordable and could be used repeatedly to follow risk assessment in these patients. We'll do an independent validation of this test as well, where we'll be collecting samples of blood, and also this will be blood samples from high-risk patients with PPNs, malignant pancreatic neoplasms, as well as pancreatic cancer patients, and our control group of chronic pancreatitis and healthy controls. And so now I want to turn this over to Dr. Chuan, who's the inventor of all this technology and has made all this possible, and I'll have him take over from here.
Thanks, Marc. As Marc introduced, my lab works on really method development. Let me just bring up the slides, because I saw Marc going to show the slides, but that's okay, I can do it. Yes, I just said we've been working with Marc to develop these tests. In addition to hydroxyl methyl C on DA, there's also 5-methyl C, as Marc introduced. In fact, there are limited number of clinical tests that utilize 5-hydroxyl methyl C and 5-methyl C. And the one that's being used clinically is actually 5-methyl C for colorectal cancer. This was the reason why we started this collaboration with Marc. So some of you probably heard of Cologuard, which actually measures 5-methyl C. Now, 5-hydroxyl methyl C is a new DNA modification. We think there's a lot of promises, but we also want to look at the 5-methyl C, which is a stable mark and has been already cleared by FDA for a few cancer early diagnosis. The major problem for 5-methyl C has been the method applied called bisulfite sequencing leads to extensive degradation of DNA. So if you draw blood, isolate cell-free DNA, it really requires lots of blood. And then because of this DNA degradation from this chemical treatment, the signal to noise has always been a major problem. So we recently invented this new method we call linear amplification-based bisulfite sequencing. It's actually a very simple idea, but it works extremely well. Now, the idea is that in the cell-free DNA, the DNA derived from tumors, small, tiny amount. What we do in this method, we linearly amplify this DNA. So from the small amount of tumor-derived DNA, it amplifies to a larger portion, and then we apply exponential amplification. So this a small amount of tumor-derived DNAs will not get lost. this idea works extremely well. In a recent test, as you can see here, with 100 nanograms of DNA with traditional method, you get very good data, right? However, with 1 nanograms of DNA, often we get about a few nanograms from several mils of blood. And with 1 nanograms of DNA, you can see the signal looks very sparse. With 100 picograms, it's almost gone. Okay. However, if you go to our method, trying to get this move. If you go to our method, as you can see, with 1 nanograms of DNA, we see very good signal, similar to 100 nanograms with current bisulfite sequencing. So this really improves the sensitivity and, most importantly, the coverage of the sequencing. So we're excited about this new method. This is the results from the recent cell-free DNA run. We applied this to pancreatic cancer patient sample versus healthy control. Again, we don't need a lot of blood samples. 1 mil of DNA, way enough for us to perform this whole genome analysis. And we have not sequenced this very, very deep to identify new biomarkers. We only did a test run. These results will come back actually yesterday. This was the reason it took us a while to put the slides together, but I want to show you the preliminary results. This is a 5-methyl-C methylation. Again, as Marc introduced to you already, we're looking at the differential methylation markers, and you can clearly see we see very different distributions of these 5-methyl-C profiles or marked cell-free DNA between these two cohorts of samples. We're excited about this. We sequence this deeper. We're going to learn a lot of not only obtain the biomarkers, but also hopefully learn the immuno and many aspects of biology from these patients. So with that, I turn back to Marc.
Yeah. Okay. Thanks, Chuan. That was elegant work. Really our goal in this whole project, and the Rolfe Foundation has been hugely helpful to help us with this, is the development of a liquid biopsy assay where we'll be able to take the patient's blood and identify early pancreatic cancers and ultimately even high-risk lesions that are not yet cancers to be able to follow over time. So currently we're identifying a panel of the most robust 5-HMC and 5-MC modified regions throughout the genome. We plan to use the next generation sequencing and the PCR-based assays to determine what the sensitivity and the specificity of the test is, and we'll use an independent cohort of patients and controls to validate the assays, and ultimately to move it forward eventually to clinical development so that we can get eventually a test that can be used widely for patients to benefit them, and that's the goal. Can you go to the next slide, Chuan? So basically, we've received already from the NIH permission to get these PLCO samples from patients who have had latent pancreatic cancers but had not yet presented, to see if our test will work for them, if we can diagnose pre-diagnostic tests for pancreatic cancer. And we're continuing to work now with Chuan on this 5-HMC and 5-MC technology so that we can get signatures that will be even more reliable than either one alone would be. And our plan is to submit an R01 application, multi-investigator application to carry this forward as well. Next slide. Okay, I want to thank you all for listening and thank you for your attention. And I guess we're open for questions for Kay's talk and Chuan and myself.
Thank you so much to all three of you. Such impressive and important information coming out of your research findings. We'll open up now for questions, and we've had some that have come through during our talk. And if you have additional questions, please continue to send them through in the chat. Otherwise, you can always email them to info@rolfefoundation.org, and we'll be sure to get to them. So if each of you can please talk about how has pancreatic cancer research evolved during your tenure? What have you seen throughout the time?
Well,
Dr. Bissonnette, let's start with you.
Yeah. from a clinical point of view, pancreatic cancer continues to be a major challenging problem because it's diagnosed late. Essentially, it's buried so deeply in the retroperitoneum that it doesn't signal that someone has pancreatic cancer generally until it invades the nerves and has become unresectable at that point. So I think we really need some way to diagnose it at a much earlier stage. And things like Ipmns are helpful in terms of CAT scans and doing EGD and EUSs to look for these lesions. But that in itself is not a good screening test. If we could screen the blood for something that the tumor was releasing at an early stage, we would have a far better chance of making a real difference in people's lives. So I think that we're still struggling with finding a good test to make this diagnosis early.
Dr. Macleod, would you like to weigh in?
Yes, no, I totally agree with Marc. I think if you look at other cancers, early diagnosis is what's made the difference in terms of patient outcomes and survival. Having said that, also, obviously, for people who are unfortunately not diagnosed early, we still need to improve how we're going to treat those advanced cancers. There's been some progress in identifying compounds that will inhibit RAS signaling, for instance, RAS being the driver oncogene. But unfortunately, a lot of these existing or those developed strategies lead to resistance after time. And so they've not stayed the course as successful therapies. I think the other approach that a number of people clinicians are taking, is combining different therapies and looking for synergy between existing therapies in treating cancers. But we really need more breakthroughs and more new ideas about how to treat the disease. And I think what's encouraging is that the public has, I think, realize that this is a growing challenge, and that there's a lot more resources going into pancreatic research, including money, but also importantly, people. I think the number of research groups working in this space has increased, and certainly in the time that I've been here at the University of Chicago, the early days when I was here, the focus was very much on breast cancer and prostate cancer, and I think pancreatic cancer, I think people sense the urgency. So I think that's positive.
Dr. He?
Yeah, for me, it's because of Dr. Marc Bissonnette, because, we always learn from our physician scientists what are the major medical problems and what kind of tools we need. Here at University of Chicago, a major benefit is we have chemists and bioengineers next door to our physician scientists and cancer biologists such as Kay. So it's really integrated, really nice system. I think some of the problems we face in clinic just require better method, and often we do have that kind of a method available. We just need to figure out ways to rebrand them and make them into the clinical applications. So I'm fairly confident in a year or two we'll have very reliable biomarkers from high-dose muscle C and phymosis.
Okay, great, because that was actually the next question was, what's the timeline that you see this being able to affect the general public? So if you have it in a year or two, how long until the public are able to benefit from it?
Right, that's a good question. So the technology, the intellectual property is owned by the University of Chicago. Actually, there is a company in the Bay Area already started. I think they got FDA approval to perform pretty wide test specifically for pancreatic cancer early diagnosis using our technology. This is very complementary with us, because I expect in a year or two, there'll be really interesting results from them and us. And we also have this new phymosis C method I'm also quite excited about. So I would say in a year or two, we'll have pretty reliable markers. I see questions some of the audience are interested. You're always welcome to come to University of Chicago maybe in a half a year, 1 year, we have markers ready.
Incredible. Do you feel that the potential liquid biopsy testing would also be helpful for screening patients who are germline BRCA1 or BRCA2 pathogenic variants?
Well, I also want to back up a little bit. To do a general screen of pancreatic cancer is economically not feasible or not beneficial. People have done that. So this type of early screen for pancreatic cancer is best to the high risk population. If I'm correct, Marc and Kay can correct me. There's such a calculation. For colorectal cancer, if you're 50 years older, then it's beneficial. But then for pancreatic cancer, it's really for high risk group, right? People with acute diabetes or with family history of pancreatic cancer. For BRCA mutations, I think that there are other ways. That's really testing of mutation. There are other very sensitive ways to detect BRCA mutation. For germline mutation, I'm actually not so sure. So I have to let Kay and Marc answer this question. So they know better.
Yeah, there's a Merck test for BRCA1, BRCA2 mutations. I think it's just a PCR test on circulating lymphocytes, or I've actually been tested for BRCA1, BRCA2 myself, and was a negative control, fortunately. But yeah, it's a fairly routine thing that can be done anyway. So
I think maybe the question was more whether the BRCA1 or BRCA2 were at risk for pancreatic cancer and should they be screened for pancreatic cancer. Is that what you were asking, Aaron? Or?
It's saying, would it be a helpful screening for patients who have germline BRCA1 or BRCA2 variants?
So I'm not an expert in this. I think BRCA1 and BRCA2 do have an increased risk for pancreatic cancer, but it's a fairly small increase. I think that family histories and other things such as new onset diabetes, for example, insulin dependent diabetes in an adult is a much higher risk factor for pancreatic cancer. But certainly I think that's a good question. And I think that any familial mutation in the germline that raises the risk of pancreatic cancer becomes eligible as a risk stratification for this kind of test.
So BRCA2 is a major risk factor for PDAC. The reason I know that is actually Hedy Kindler here has got a clinical trial running for combining the use of using PARP inhibitors for pancreatic cancer, based on the fact that there's a subset of patients that do have BRCA2 mutations. It seems to be mainly BRCA2 for whatever reason, not BRCA1.
Okay, thank you. Our next question comes in saying, I have 4 members of my father's family that passed away from pancreatic cancer. I'm 57 now. Will these tests be available soon, or what should be my first steps to somewhat protect myself?
I think we're going to have some data in a half a year, 1 year, but then to really get this into a clinical test is a long journey, right? Different layers of steps we have to. But yeah, you're welcome to contact Dr. Bissonnette and do blood as a volunteer. I'll be happy to include into our studies, and that we can always compare those samples with our data and have a risk evaluation. I'm not sure if that's the correct answer, Marc.
I know. I think that's one approach for sure, and we'd be happy to do that. The company you mentioned out in the Bay Area, is that Celera? Is that the company?
Blue Star Genomics. They're using this hydroxyl muscle C. Remember?
So would that be another place that you might have her blood tested? I don't know.
Well, probably not there, but they can always come here. We can accommodate a few, I guess.
We're happy to do that. So,
isn't there a clinic? Doesn't Sonia Kupfer run a cancer risk clinic for?
She does.
Yeah.
I don't think she's focusing on blood tests for pancreatic cancer, but she does have a high risk cancer clinic and could be seen in that clinic, and we could certainly collect the blood and analyze it. Yeah,
I think all of these folks are obviously anxious and want some good screening to make sure that they can do what they can. Our next question has to do with your thoughts on the newly approved IMMray PanCan blood test versus what you're working on. What can you elaborate or talk a little bit about what the IMMray PanCan blood test is, if you know, and how, if it varies from the work that you're doing right now?
Chuan, I'll defer to you. You probably know more about that test.
I actually don't know much about that test. Is that a cell-free DNA test that come out of Illumina, or it's a different one?
That I'm not familiar with. We could come back to that one. So how early are you anticipating, like stage-wise, pancreatic cancer will be able to be detected in the blood work?
Well, that's a great question, and we don't have an answer. I mean, we're looking, in the case of colon, we have advanced adenomas, advanced polyps that we're seeing signatures from. And in our preliminary data, it looks as if the more advanced dysplastic ones have a different signature from the less advanced, less dysplastic. But until we have larger numbers, Aaron, it's really hard to say. It's a work in progress. I'm hoping that the 5MC will actually add a lot to this story. I think that it will. I'm encouraged by already what Chuan has done with 5MC to suggest that that may give us earlier signals and more robust signals than just 5HMC alone.
Dr. Macleod, this one's for you. I know that there's not a confirmed successful treatment of cachexia. Is there any biochemical support for fish oil and increased protein to help ameliorate the progression of muscle degradation?
I'm not aware of any work with fish oil or omega-3. I mean, increased protein. I mean, Marc's a physician, might and know more about this as well, but my understanding from the literature, and certainly our mice as well, is that increasing their diet and increasing protein can help them, nutritionally, stay alive, but it does not prevent the cachexia. It does not reverse the cachexia, and it doesn't it doesn't stop it. So that I know from a number of patients that I've actually interacted with on this topic that they cannot eat their way out of cachexia, unfortunately. But it can obviously provide energy and nutrition for the brain and other tissues, but it doesn't stop the cachexia.
That's my understanding as well, Kate.
So I know we're coming at this from a research perspective. I'm curious, though, to see if you have thoughts or knowledge on how this early detection is going to affect treatment for pancreatic cancer patients.
Well, that's another great question that I don't have an answer to. I mean, I think that in the chemoprevention literature, there have been many trials in colon and mostly not giving us much guidance. I mean, aspirin has gotten a lot of play, and there is still a lot of interest in aspirin in terms of its ability to prevent colon cancer. But other things such as vitamin D and a number of other agents, fiber in the diet, et cetera, have not. My own feeling is that until we have a reliable test for early lesions, we can't really address whether we can prevent it. And of course, the clinical dilemma of whether to do a Whipple procedure or not is a huge one. I mean, it's a huge operation, and in patients who are at high risk, of course, you can sample the pancreas with fine needle aspirations, and if you get malignant cells and you stage them as resectable, then there's no question they should have an operation. But in terms of deciding, what to do at a stage where they're not yet advanced, I think that remains a big conundrum clinically.
And so from listening to the three of you tonight, it sounds like the history piece of it is pretty big. And so those who are not at risk at all, where it comes out of the blue, if you will, that is there any research for that type?
So that's the sporadic pancreatic cancer as opposed to familial. And that's most pancreatic cancer, in fact, are sporadic. There are people that comes out of the blue. They don't have a family history. And my hope is that a blood test, there are a number of blood tests that are being done now that are sort of pan-oncogene tests. They're looking at many cancers. They're not focusing on just colon or just pancreas. And my hope is that maybe the 5-HMC, 5-MC story or something like that will serve such a purpose, that we'll see the detection of cells entering the bloodstream which are abnormal in their signature. They don't look normal by DNA signature, and that will set off bells to at least begin to investigate that. Whether the problem right now is that imaging is not very sensitive, so that if you had abnormal cells and you did a CAT scan and it was negative, and you did an endoscopy and an ultrasound and that was negative, what exactly do you do at that point? I think the most you could do is probably repeat the blood test on a regular basis. And if it looked as if the signature was good for the pancreas and the numbers were going up, you might have to make a very difficult clinical decision as to whether or not to operate.
Any other additions, Dr. Macleod or Dr. Hay? Okay.
I agree. I think the ultimate goal, this is the reason why hydroxyl muscle C is interesting, because we perform whole genome sequencing. So in principle, one can look at the colorectal cancer, ovarian cancer, breast cancer, and pancreatic cancer in one experiment, or maybe more cancer types. For 5-methyl C similarly, but it's much harder to do whole genome 5-methyl C. There, the panel-based approach might be feasible in the future.
Dr.
I just look at this Emory test. It's a CA19 test, so it's still protein-based, but they probably expanded with other biomarkers. So it's still different from the test that we're trying to achieve.
It's interesting, the CA19-9 was used on the PLCO samples and actually had some success in identifying patients who had not yet presented with pancreatic cancer, but were found to have an elevated CA19-9 in their in their early stages, but prior to at a time when they were asymptomatic and had no evident cancer. So my sense is that the test that Dr. He is working on is going to be much more sensitive than the CA19-9. And he's already published studies showing that the 5-HMC signature can identify differences between, for example, liver, pancreas, lung, ovarian cancers. So I agree. I think that this could be very promising for looking across the board at many different tumor types.
So Dr. Macleod, you had touched upon this in your presentation, and I wanted to expand upon it. So the notion that pancreatic cancer receives so much less funding than other cancers when it has such a low survivor rate. Why do you think that is? And you had mentioned the evolution of that. Can we talk about that a little bit more?
Yeah. So it has changed. I mean, I think historically, breast, prostate, obviously lung cancer, colorectal cancer were more prevalent. And to some extent, it's understandable that more money did go in those directions. But again, I think give PanCan and people like Lynn Matresian a lot of credit for pointing out that by 2030, that pancreatic cancer will be the second deadliest cancer after lung cancer. And I think that statistic has woken people up. It has made people realize we've got to do something about that. There is one angle to it that we haven't really touched on, and that is, why is the incidence of pancreatic cancer increasing? We can talk about diabetes and obesity, but we really don't have a good scientific handle on what is causing the increase in incidence. But as I said, again, there is more and more money going into pancreatic cancer research. It's not just NIH and NCI. Just in the last year and a half, the Department of Defense has set up a pancreatic cancer specific set of funding mechanisms. So I think it is changing, and hopefully all those additional resources and people researching it will make a difference. It's made a difference in the other cancers where that's happened. So one has to be optimistic.
Dr. Bissonnette and Dr. He, do you have anything to add on that notion?
Would second everything Kay said.
So bear with me on this.
I think Kay just built a pancreatic working group here on campus. They're meeting every other week. So we're making lots of efforts here on campus already.
Yeah, we have a pancreas cancer working group that started actually February 2020. I think the first meeting was the lab when we met with you the last time. It was around that same time. And it's been going strong. Our new cancer center director has identified pancreatic cancer as an area that he wants us to develop more effort in, and we are going to be pulling together a SPORE grant next year. So that's a very translational effort. It takes exciting new scientific discoveries and make sure they get into the clinic and that they do have an impact for patients. So I think here, I mean, Chuan's right, at the University of Chicago, I think we're particularly well placed to have an impact. Our surgeons are actually amongst some of the best in the world. People like Jeff Matthews and Kevin Rogan. And so that also gives us a resource that not other places don't necessarily have. So I think the combination of the great clinicians and the exciting basic research like Chuan's work and others, I think it's exciting. Obviously for a patient, the sooner we can get this together, the better. Time is of the essence.
Absolutely. So bear with me on this next one, because there's not a great direct correlation here, but I still want to make the analogy. We got the COVID vaccine so quickly because researchers from around the world partnered together. They shared findings and basically did whatever it takes to make it happen. Why don't we see that with pancreatic cancer or any cancers for that matter?
I think you're right. I do think that we do work together. I mean, I just mentioned the pancreas working group. I think PanCan is an example of lots of different researchers in pancreatic cancer sharing data before it's published and collaborating and not necessarily just duplicating each other's work. So I do think that is happening. I think the issue is that pancreatic cancer is a much more challenging problem to solve. I mean, hard to say that compared to COVID. I mean, COVID it's a virus. You've got a very obvious target. You've got a coat protein that you can raise an antibody to, so that it's an easier target. It's an easier solution. I mean, obviously, a lot of work went into that, but pancreatic cancer is much more challenging.
For sure, and by no means am I trying to minimize your work and the everything that is doing. It was more the partnership and working together. So what can we do? What can our community do to help?
I just want to add a few more comments following Kay's comment. I think basic biology is really the key for COVID. From basic biology point of view, it's not really a challenge at all. It's just a matter of time, because for the last half a century, the community have built so much basic biology on virus. If you really want to think about the hard virus, HIV will be one, right? Because COVID and flu will be another one. COVID is a tough virus, but in terms of basic biology, we know so much, and there's so much tools being developed. Pancreatic cancer and the cancer in general is such a major beast that intimately linked to human developmental biology, everything. We just don't have enough basic biological understanding. As Kay beautifully showed that this muscle to pancreas communication, we don't even know the signals, right? Just so much new science we need to learn. I think we can make some immediate impacts potentially by developing biomarkers, but to really fight or combat this cancer is going to be a long journey, as also Marc indicated. Early on, you can perhaps do section, but then the later stage, how you really deal with it, it's going to be challenging.
Sure. Thank you for that. So what can our community do to help? We're all out here, we want to help you and help our family and friends. How can we do that?
Well, I mean, again, without being too brutal, I mean, research is expensive, so the funding is really critical. I think one thing that, support from an organization like Rolfe allows us to do is to do things that are perhaps a little bit riskier and a little bit, wouldn't necessarily get funded at NIH, in its current form, but allows us to develop new ideas so that down the road we will get funded to continue the research. So I think that's where the funding is really very, very useful. I think if it's not an aspect of it I'm involved in, but I know, people donating samples to tissue repositories, and if you are involved in or want to become involved in a clinical trial, I know that our clinicians are always looking to enroll new patients in clinical trials, if even as a control subject. So that's something, not everybody is going to want to do that, but that's something I know our physicians, again, Marc probably can speak to that better than I can.
No, I think that's a really important thing. I think that advocates of particular, problems like pancreatic cancer, by virtue of their family histories and their own stories, can provide an invaluable resource in terms of the blood that we're trying to make a biomarker from. So it's an important contribution, a huge one.
And what do you expect to see in the days, months, and years ahead? Or I'll combine that with what are 3 takeaways, maybe one from each of you, that you would like our community to leave here with tonight?
I think that there will be a biomarker for early pancreatic disease that will make a difference in this disease, and that's what we're working to find.
Yeah, from our perspective, I think the idea that at least if we can mitigate tissue wasting, muscle wasting, that we can at least extend lifespan for patients. At the end of the day, we'll almost probably also need new therapeutics. So I think those, the early detection, new therapeutics, and understanding ways to extend lifespan and make patients more responsive to the new therapeutics, I think will be really, really helpful.
Dr. He?
Yeah, I will add that I think a very quantitative detection and analysis of a variety of biomarkers from pancreatic patients will tell us a lot about the tumor microenvironment, immune modulations, as well as why patients respond to certain therapy better than the other therapy, or some patients do not respond to certain therapies. And number one, that will help hopefully in the future clinical treatment, but also provide the information for basic biology understandings, and eventually leads to better treatment.
Great. Well, I want to thank you so much, Dr. Bissonnette, Dr. He, and Dr. Macleod, for sharing such important research and findings with us tonight. It does give such hope for what's ahead, and I just want to thank you for sharing with us tonight, and we look forward to seeing what's to come.
Thank you all very much for listening.
I also want to thank everyone for joining us tonight. If you're catching the replay, you can go ahead and write replay in the comments with your question so that we can get back to you after we ask the doctors. I'd like to close tonight with something that Rolfe's executive director, Stacia Hart, said to me during our conversation this morning. We were talking about the events of the past 24 hours, let alone the past year or two. And Stacia said, we're family and we're there no matter what. And it really hit me that she's right. That's what makes the Rolfe team, the community, so very special. It's the connections and the support from institutions like University of Chicago, our staff, our supporters, patients and their families, and that make this community one of a kind, truly. The determination and the fight that we see from and with each of you, and the often gut-wrenching vulnerability and willingness to simply show up for those who need us. On behalf of everyone at the Rolfe Pancreatic Cancer Foundation, we thank you so much for your support, and we send some love and light to you as we continue through this holiday season and into the new year. Until next time, stay healthy and take good care. Good night.